بسم الله الرحمن الرحيم. Assalamu alaikum wa rahmatullahi wa barakatuh. Our lecture is about gene mutations. The objectives of mutation lecture are: define mutations and describe how each type of mutation can cause a phenotypic change. List and distinguish between the different types of mutations that result in phenotypic change. What's the meaning of mutation? Mutation is changes in the nucleotide sequence, generally showed up as altered phenotype due to changes in the genotype, usually happened during the DNA replication or due to chemicals or radiation to the DNA molecules. May occur in gamete, egg and sperm, and be passed to the offspring, or may occur in the somatic cell, aren't passed to the offspring. The gene mutations can be classified into three types according to their effects: either beneficial mutation, neutral mutation, and harmful mutation. In beneficial mutation, this type of the mutation produced a new genotype whose phenotype may be better adapted to their environment. For example, sickle cell anemia. Sickle cell anemia is one example on beneficial mutation. Hemoglobin mutation, called HbS, that makes red blood cells take on curved sickle-like shape. With one copy, it confers resistance to malaria HbA/HbS. But with two copies, SS, it causes the illness of sickle cell anemia. يعني الشخص الحامل للAS في نفس الوقت يكون مقاوم للملاريا ومقاوم إلى مرض السيكل سيل أنيميا. This image shows the normal hemoglobin and this sickle hemoglobin. The other types, called neutral mutation. Any changes in the gene sequence cause changes in the structure of the proteins. These mutations do not show any advantage or disadvantage to an organism, such as red hair. We can see the person with red hair does not gain any benefit or advantage as the person with the black or blonde hair. The third type of the mutation is harmful mutation. Many mutations are harmful because changes in the amino acid sequence of the protein, for example, thalassemia or called Colles anemia. Thalassemia is a hereditary anemia in which synthesis of globin polypeptide alpha or beta chains are reduced or absent, that it is leading into the production of the very low levels of the globulin. There are two types of thalassemia, according to which of the globin chain is absent, either alpha or beta thalassemia. According to the chemical changes, there are three types of the mutations: base substitution, base deletion, and base addition. Base substitution, or called point mutation, in which one nucleotide is replaced by the another. Some of the point mutation arise due to spontaneous errors during the DNA replication, and the others are result of damage to the DNA caused by mutagens such as radiation or chemicals. There are two types of base substitution, either transition and transversion. In transition, the pyrimidine changed into the another pyrimidine. So, for example, cytosine into the thymine, or purine changed into the another purine. For example, guanine changed to adenine. The other type, transversion, in which the purines and pyrimidines are interchanged. This type more rare than the transition. There are many types of point mutation. Silent mutation. There is no noticeable effect because both of those codons code for the same amino acids. For example, if the CCG is changed into the CCC, both of those codons code for the same amino acids called proline. Other examples, if the C or cytosine changes into the thymine, then transcript into the messenger RNA and translated into the amino acids. Both of the amino acids glutamic acid. The other types called missense mutation, in which the single nucleotide changes that cause the substitution of one amino acid for the another in a protein. This substitution may or may not affect the function of the gene product. For example, if the CTC code for the glutamic acid changes into the CAC code for the valine, result in sickle cell anemia. What if this DNA was changed to this DNA? What does it matter? CTC codon for glutamic acids and CAC codon for the valine. What does it matter? The normal hemoglobin was changed into the sickle hemoglobin. In sense mutation, they produced longer than normal proteins by changing a stop codon into one that code for the amino acids. Nonsense mutation, in which they change codons that specify amino acids into the one of the three stop codons, UAA, UAG, or UGA. That leads to the shortened polypeptide chains. That it is contrary to what we have seen in the sense mutation. In sense mutation, leading to longer polypeptide chain instead of the short polypeptide chain in the nonsense mutation. For example, blood clotting disorder changes the GAA coding for the glutamic acid into the UAA coding for the stop codons, lead to the blood clotting disorder. That means cause the blood do not clotting during the surgery or injury. The blood clot disorder occur if the GAA on messenger RNA codes for the glutamic acid changes into the UAA, that it is lead the stop of the translation and cause blood clotting disorder or hemophilia. The other types of the base substitution, it is called splice site mutation, in which can change the phenotype if an intron is translated into the amino acids, or if an exon is skipped instead of being translated, that it is lead to shortening of the proteins. كما عرفنا سابقا بأن الجين يتكون من انترونز وإكسونز، يجب أن يحذف كل الانترونات الموجودة على الجين وتبقى فقط الإكسون اللي هي transcript and translated. في حالة أن الانترونات أو أحد هذه الانترونات transcript and translated into the amino acids راح تعطينا فينوتايب مختلف عن الفينوتايب الأصلي للبروتين الأصلي، أو في حالة أن الإكسونز اختفت وبدل ما تترجم توقف عملية الترجمة، راح تؤدينا بهاي الحالة إلى قصر بالبروتين أو البوليبيبتايد الناتج. For example, cystic fibrosis. In cystic fibrosis, an intron site is not removed. Instead, that is translated. The encoded protein is too bulky to move to its normal position in the plasma membrane, where it should enable salt to exit the cell. As a result, chloride accumulates in the cells and water move in, causing drying and thickening of the mucus outside. إذن سبب السيستيك فايبروسيس هو حدوث طفرة أدت إلى عدم حذف إحدى الإنترونات، والتي بدورها ترجمت إلى بروتين. هذا البروتين مختلف عن البروتين الأصلي الذي يعمل كوسط ناقل لتبادل الأملاح ما بين داخل الخلية وخارج الخلية، وبالنتيجة أدت إلى تجمع الكلورايد بداخل الخلية وحركة الماء من الخارج إلى الداخل الخلية نتيجة لاختلاف التركيز. That it is cause drying and thickening the mucus outside. The other type of the gene mutation is base deletion, in which involve the elimination one or more bases. For example, many cases in the male infertility are caused by the tiny deletions in the Y chromosome. The third type is called base insertion or addition, in which involve addition of one or more bases in the gene. For example, in Gaucher disease, an inserted single DNA base prevents production of an enzyme that normally breaks down of the glycolipids in lysosomes. So, the build-up of the glycolipids enlarges the liver and spleen and causes easily fractured bones and neurological impairment. The deletion and insertion called frameshift mutation that can be produced completely non-functional protein because the sequence of the codons completely altered. In this sentence, the fat cat ate the wee rat became meaningless if the A is added to it. In other example, if the G base eliminate from the sequence, that it is lead to the change of all the polypeptide. The other type of the gene mutation, it is called inversion. Inversion mutation only affects a small part of the gene, in which small part of the gene is inverted. For example, if the TC inverted into the CT, that it is lead to the changes in the polypeptides due to changes in the amino acids glutamic acid into the glycine amino acids. This image shows different types of the mutation. This animation shows different types of the gene mutation. Thank you very much.